for joining us and happy Science Week. I hope you've all enjoyed some really great activities so far. I'll just introduce myself. I'm Jenna. I'm the Training and Outreach Coordinator here at the ALA. And I'm joined here with my colleagues, the wonderful Kylie, who will be helping with our comms and tech. And we have the wonderful Margot, who's going to take over from me and give us a live demonstration of the platform after I give a few slides. And we have Tim, who's our resident user support officer, and will be able to answer all your questions in the chat. So as we go along and you have burning questions, put them in the chat. The team will try and get to them straight away. And if they're relevant for the rest of the group, they can just interrupt me or Margot and we can answer those as we go. Okay, so let's get started. So before we start today, I would like to take a moment to acknowledge the traditional owners on the lands that we're meeting today. And this includes the Ugambe people from Currumbin Valley, where I'm sitting today, as well as the Ngunnawal people of Canberra, where my colleagues are working from. I'd like to extend my respect to all Aboriginal and Torres Strait Islander people, including elders past, present, and emerging, and recognise their extraordinary contribution to the science and the data and the work that we'll be discussing today. Okay, not sure what this one is, sorry. Okay. So let's get started, and I'm just going to introduce what the ALA does. So the Atlas of Living Australia is essentially a one-stop shop for Australia's researchers and decision makers to access trusted biodiversity data. And our data is key to answering lots of important questions in conservation biology. For example, we can help in understanding and managing species and ecosystem changes. We work with and provide data for leading experts in biosecurity, and this helps them to understand distributions and impacts of invasive species. We also support data-driven species identification, description, and discovery, which has a big impact on Australia's taxonomic research. And our data enables effective ecological restoration and management. The ALA is Australia's biodiversity data infrastructure. So we really pride ourselves and the platform on being collaborative, open, accessible, and usable. The ALA's digital platform allows you to access species information. This includes things like descriptions, names, classification, literature, images, as well as over 100 million species occurrence records. It also allows you to download this data, so either using the digital platform, or we also have an R package called Galar, which some of you might have joined the workshop yesterday. We allow users to map and analyze data on the Spatial portal, and also collect, upload, and share data with us. And we're going to show you some of these different tools and functions in the demonstration after these slides. So currently the Atlas has over 100 million species occurrence records, of which nearly all are open and freely available for you to use and access. And the ALA also shares its infrastructure with over 27 countries and has over 750 different partnerships. Our data facilitates incredible research and has been used we're up to 4,000 scientific publications now, which is amazing. So as part of our goals are to be collaborative, accessible, and open, we share and we link our infrastructure to multiple different nodes. So these include Digivol, which is an online tool that enables volunteers to capture data and to digitize collections that are held within museums, libraries, archives, and herbaria. We also connect with iNaturalist, which is one of the world's most popular nature apps for recording and sharing species observations, and the Biodiversity Heritage Library, which is a database for biodiversity literature, and of course GBIF, which is the Global Biodiversity Information Facility, of which we're the Australian node. So let's now have a talk about the data that we have on the ALA. So how we source it and how you can access it. We have lots of different types of data here on the Atlas, but the majority of that data are species occurrence records, which are records of a species that are located at a certain time and a certain place. These types of records are widely used in the scientific community, and our stats suggest that species distribution models are one of the most common analyses that use that data type. Our occurrence records are collected in lots of different ways, and you can see that in the diagram here. So for example, some of our data is collected as preserved specimens from museums or herbariums. And for this kind of data, we have around 14 million records. Or more commonly, we have data that's collected as human observations. And this actually accounts for around 82 million of our occurrence records. We also host some genetic data and are currently working on exciting collaborations that will increase the visibility and usage of genomic data. We're also constantly working on our tech so we can enable new and exciting data sets. So please watch this space as we continue to kind of get more different types of data and increase our collections. So as well as having species occurrence records, we also have other data types available. So for example, we have species information such as taxonomy, conservation status, Indigenous species names, and published literature. We provide spatial layers that can be used for analyses. For example, we have both environmental and contextual layers available. And we have lots of different tools that you can use to model your data. For example, we have tools on the calculation of species distributions, endemism, and abundance. So this here is an overview of the occurrence data that we have on the ALA, which currently is over 100 million records. So you can see that there are some trends on this map that you can see here. So this is sectioned off by IBRA and IMCA regions, so different biodiversity layers. So some trends you can see, they largely follow trends in collection. So we have lots of data collected on the southeast of the country here, but we have less in the middle and a bit less here in the north. So this largely follows trends in species richness, but also can reflect accessibility for surveys and fieldwork. And on the right here, you can see the trend in our collection of records through time. So the collection of species record data really started to increase more rapidly since the 1970s and continues steadily, particularly for human observations. Museum specimen collections do start to plateau, particularly since around the early 2000s, which is kind of expected as there is a limited capacity in those sort of collections. So since the collection of our data really took a spike since the 1970s, we've produced this graph here of all records collected since then. So you can see here that the majority of our records are birds. So we have nearly 50 million records of birds collected since the 1970s. This is followed by plants with a lot less, but still at 15 million records, and invertebrates with around 4 million. There are fewer records for fish, mammals, reptiles, amphibians, and fungi, but these still continue to increase over time. So how do we actually get our data? So the ALA is a data aggregator. So this means that we don't actually collect the data or own the data ourselves, but we rely on the scientific and citizen scientific community to share that data with us. So we have a range of key data providers that share their data with us. Some examples include Frog ID, eBird, BirdLife, iNaturalist, along with lots of museums, herbariums, and research institutions. So how does it work? So the data is shared to the atlas in a Darwin core standard. So this is a format that really just has standardized fields in a CSV file, and they include things like date, ID, species, and location information, along with quite a few others. So this data is sent either online, through email, through the platform, or it's automatically ingested by our data management team. And we also have some automated processes that help identify issues with the data, such as missing information, but we also do rely on the accuracy of the data submitted to us. Data quality is a big priority for the ALA, and we've developed a data quality project which allows the Atlas to continually improve our data quality and vetting capabilities. So we now do this by performing a few things. So for example, we perform technical and consistency checks, geographic and time checks. We check for species distribution outliers. We make sure that all data quality checks are visible to users, and we can show that in our demo. We also notify data owners if their data has been flagged with any potential issues. So for example, if you notice an occurrence record, for example, that you know it's maybe a desert species, but it's occurring in the ocean or there's something clearly wrong with it, you can actually lodge that issue with the ALA by clicking Flag an Issue. So that's this one right here. So you can fill in this issue, um, which looks like this. So you can select what's actually wrong with that, so the issue type, give us some more information in the comments, and you can also be notified when we've actually processed that. And we can show that in the demonstration. The ALA also has data quality profiles. So these profiles easily allow users to remove what we call suspect records from their intended data set. So some examples of this include removing records with spatial flags, having potentially naming issues, so the wrong scientific name or spelling mistakes, and duplicates, among many other different checks that you can see here. So these are the profiles that you can use to enhance your data quality, kind of as a first sweep. So when you actually download that data, it's already excluded these ones. And you can see here that each different profile, which you can easily turn on and off by clicking the checkbox, it excludes a certain number of records. And again, it's really easy to turn on and off based on your needs. So I'm going to move on now, and we're going to start to move into our demonstration section. So before we get started on learning how we can search for species and data, we first need to have an account logged with the ALA. So if you already have an account, please sign in if you want to follow along. If you're happy just to watch, that's no worries as well, no worries at all. Or you can jump on and create an account. So it's free, free to use, and we definitely don't spam you with loads of emails, so no worries there. So in our demonstration today, we're going to walk through a few things, including how to search for species records, how to apply alerts, data profiles, filter your results, download those records, and we'll also do a quick tour of the Spatial portal and the Explore Your Area tool. And I'm going to stop sharing my screen now and pass over to Margot, who's going to give this demonstration for us.
Hey everyone, so I will share my screen. 2 seconds. All right, so we should just be at the ALA homepage now. So we'll go nice and slow so that everyone can follow along. And any questions, just pop them in the chat and we'll answer them either via the chat or I can show you if you get lost. So we're going to start with a genus. So we're not going to go down to the species level, but we're going to look up Taliqua, which is a lizard. So you'll notice that when you search it, lots of different options are going to come up on your screen. And in this case, we just want that first one. So it's tried to give us, that list will try to give you everything that's related to the name that you've searched. So you'll get all of the different species. But at the moment, we're just going to go to the genus level. So we're going to click on that one there. So this is a species page that we've got up, and we've got some really beautiful photos of different types of Taliquas, and we also have the indigenous name for the species in the Wajari language, which is lovely to see as well. So when we scroll down, you'll see you've got brief summaries, and so this stuff's taken, this is from Wikipedia, and then also Encyclopedia of Life will have some information. So this is just a brief overview taken from these web pages. On the left, and some photos, and you can go through the tabs and see more photos. You'll get specimen photos, which aren't always the prettiest, and then images also submitted by the general population. And you can always scroll through those if you want more information on old names and taxonomy of this genus. So in this overview, you can also, let's say you're particularly interested in the Taliqua genus and you want to know more about it and you want to know when information is more species are recorded and more people upload records about this. So we can click on this receive alerts when new records are added. And that's going to take you to a page like this. And you'll notice if you haven't logged in, you'll be prompted to log in at this stage. So if this doesn't work, it might be because you don't have an account, but that's okay. So we can go through and you can customize what you want on this page. You can do it with lots of different things. So if there's something that you're particularly interested in and you want to know more about it, then this is where you'll go. And you can tally things on and off depending on how much information you want. So we'll just go back to the species page. The next thing we're going to do is we've got this map here and we want to know what this map is actually showing us. So there's lots of different individual dots on here, and we're going to click on the View Records button on the right, which is going to take us to what's called the biocache. And now this has different information, but that was displayed on the species page. If we scroll down, you'll notice there's lots of different things, and so these are all the different records for this species. It's taking us to a map, okay. Go back to records, so we can click records. We can also do map, and there's more information on those tallies as well. So I'm going to stay on the records portion of this for the moment. And we're going to go through some of the data profiles, which is what Jenna was talking about just before in that presentation. So you'll notice just at the top here that we have something that's called data profile. Now the default is going to be ALA general. So you'll notice that we've got this set, and this is to deal with suspect records. Because we get records that are submitted by the general population, and you also get records, things don't always go according to plan. And so some are going to be, I'll go to the map to make this a little bit clearer. And you'll notice here that not everything has been removed because the records, the way we filter these records isn't always perfect, and it can be quite difficult to deal with. But this first one excludes spatially suspect records. We've removed things that are absence records, so where someone has identified that something isn't present rather than presence, and scientific name quality. So let's say someone's inputted a record and it doesn't quite match to the species or the genus that we're looking for. I'll zoom in on that as well to make that a little easier to see. There we go. And also duplicates. So sometimes we get duplicates from some of our different providers, and you don't necessarily need the same record multiple times. So you can see that's removed quite a lot of records as well. So that's the most commonly used one, and that can be great if you just want to, as a base filter, remove some records. But if you want a little bit more customization or other filters, besides ALA general, we have species distribution modeling, which has less layers, and I'll zoom in. And so it excludes duplicates, and you'll notice here that that's a much bigger number one than before, and that's just because the standard for duplicate and the standard for spatially subset records is different for species distribution modeling versus our general filters. So those numbers are going to be different, and you can investigate that a bit further if you want by clicking on the little icon, and it'll tell you how this stuff is calculated. So if you're ever uncertain how these things get sort of calculated through, you can always click on the little icon buttons and you'll get lots of information that way as well. And same with the filters. So you'll notice species distribution modeling, and then there's lots of information, and some of these links will take you to Atlas GitHub pages if you need to know the code and everything behind that as well. So we'll close that there. And then we also have, so data license for all uses. Some data isn't, so if you want to make sure that if you have to share anything, that you want to use this one. This one is Australian Vegetable Herbarium, and then you can also disable the data profiles if you don't want them. So this will now show everything. It's hard to tell with a genus of this many species and this many records that when you disable it, things come back. But if you're looking at something that's a little bit more specific, then you'll have a clearer idea that things have been removed with these data profiles. So I'll just turn that ALA general one back on just because that's the standard one. And from this, so these are our general profiles, and it's a group of filters that have been made to make these profiles. So these are all individual filters, And what you can do is you can also customize your filters. So I'll zoom in again to make this a little bit easier to see. And we'll go to the left. So underneath that occurrence records, you'll have customized filters. So you can click on that. And then we've got all these different things that you can click on if you want to filter them by. So you can filter by taxon, by identification, by occurrence, by location. So a good one as well, and then all the way through. So state and territory, you can filter by country and everything like that. So a bit unwieldy on the scrolling, sorry. Scroll back in around here as well. So if, for example, you just wanted a specific species of Teliqua, you could click on this one here, Teliqua rugosa, and now you just have those. So we've removed all other species of Teliqua and we're just left with this singular one as well. So that's can be a good way to navigate if you're interested in just a specific species inside. And you can also do that with other things as well that aren't just a species. Just scroll back over here. We can untoggle that so we get back to everything, and then also scroll down. And you can also filter by sort of institution is another one if you want to know who's provided us with the most records for this genus, and you can scroll all the way through. And so here we've got South Australian Museum is actually something that's provided us with a lot of records, and based off of the name of South Australian Museum, you'll realize that most of those records are also in South Australia. So that's logical, which is good to see. So we'll scroll back out. And from this, we're going to move on to a record. So we're on a page that has many different records. I might just untoggle that institution one. Great, so we're back to the home page. We've got our ALA general data profile on. So now this would be exactly where you would come to if you had just come off the species page and clicked view records. So we're back to just from about 5 minutes ago. And we've got all our records, and because we're at the genus level, you'll see that there are different species in this list. If you were just looking at a species, then only a singular species would appear here. So we're going to go and we're going to click on a record now. So let's go with this one. It's a Eastern Blue Tongue, New South Wales, and it's a human observation. So this is something that someone submitted by like a general person, just like one of you guys, has gone out and found it and submitted it through iNaturalist, and then we've gotten that data. So I'm going to click on View Record, and we even have some photos with this one, which is nice, and you'll see them on the left, which I'll just zoom in on nicely for you guys. Quite a large lizard as well. And so there's lots of different information on this page, and it can be a little bit overwhelming, and it's just to supply everything that all the information we've received, and then all the information, sort of everything that we've done to this record to get it to where it is that you can see it. So from here, this is the standard information that you might be interested in knowing. It's an iNaturalist data. If you were to follow that link through, it would take you to that original submission and it would take you to iNaturalist. You go through, this is a present record, so it was there and there's a photo of it, so that makes sense. And then we'll scroll all the way down. You'll notice this is its scientific name. It's been identified as a species. And then we'll just come all the way through, and you'll realize that there's also lots of quality tests that we've done on these. So it's passed 94, so 94 passed properties, where it's missing one and some haven't been run. So this can be a little bit confusing, but it just means that when we check for things, so it's not a record that's in the ocean, it's not a spatially suspect record, it matches the taxonomy correctly, so it is what it says, the picture matches. It's all things in that realm to make sure that we're providing you with a record that makes sense. And if it isn't something that has passed, we show this information here and you can make a decision as to whether or not you would like to use that record or not if you need it for a specific thing. So you will notice that on one of these things, it's a warning. So it hasn't been removed, but the coordinates have been rounded. So it's potentially not as many coordinates as it was provided. and it's slightly less precise than it was originally. So that'll come up with a warning here. And every single occurrence record will have this, and there's lots of information to explore on everything. So you'll notice this is what it means by coordinate rounded. I'll zoom in a little bit so you can see, is that the person supplied us with quite a few points after the decimal, and we've rounded that up to that point there. So that's all that's telling you, is that we've modified the data that was originally supplied to us. And again, if you scroll down, there's lots of other information on here, which I won't go through because there's a lot of it. So we won't do that right now. What else do we want to do? So let's say, so this record is okay, but let's go back and let's go to the map. Now, we're looking at lizards, and there's a record here in the ocean. So we're going to go view record. Now, to me, and it's pretty clear that there's a problem with this. It's sitting in the middle of the ocean. So what Jenna was referring to before about flagging an issue. So if you notice something like this, where it's really clearly not correct, you're going to click on flag an issue. Whoop, it's popped up in the right-hand corner. And you've got different options here, which if you think it's a duplicate, temporal, taxon. So this isn't a taxon, but it's a geospatial issue. So you can say record in ocean and submit that through. Great. So now that record has been flagged, and it'll be spatially suspect when someone else goes to look at it. And if someone wants, the person that submitted this record will be notified that there's a problem with the record that they've submitted. So from here, we're very zoomed in. Okay, so we've got everything of the genus Taliqua, and let's say you wanted to do some research on this, or you're just really interested in having all these records, for whatever reason that may be, you can now download all of these records from this occurrence page. So from the bio case, we're going to zoom in a little, and I'm just going to go to the right. I'm just going to click on this download button. That's going to take you to this page, and you're going to have a couple different options here. So you can select occurrence records, which will give you all of those records in a CSV file, and you'll have lots of different columns. So you'll have the genus, you'll have the species, you'll have the kingdom, you'll have everything, you'll have pretty much more information than you'll need on that. And so you can just take stuff away as you wish. You can also get a species checklist. You can also get a species field guide, which will come with some photos and some things if you ever wanted to go out and look for this species, maybe in your backyard or something like that. And so I'm going to click occurrence records on this one, and then you have a little bit of discretion as to what you want to do. So you can name it, download format. Just going to keep it on the auto ones. Again, if you're unsure, you can just click on the little question marks and they'll take you through to other things. And then output file format, CSV is the standard one, but you can also do it as a TSV as well. So that's what you would do for that. And then from there, we need a reason. So we'll just say testing, and then we'll click next. I'm not going to download this right now just in case it takes forever and then we're pending, but that'll just get downloaded and it'll get sent to your email as well. So you'll receive that in an email, which means that you can track it. That's great. So I'll just go back because I don't want to download that, and we'll zoom back out. From here, we're going to do a quick tour of the spatial portal. So if you want to do more visualizations than just having sort of this map, which you can zoom in on and you can click on records, but there's not that much more that you can do on this map. If you want something that's a little bit prettier and has more power to it, we can click on this button, which is View in Spatial Portal. You'll also have noticed that from the species pages, you can also click on the spatial portal from there. So you don't have to go through this. I'll just show you that now as well. actually. So if we go back to here and we were to research that species, that genus, sorry, and click in, this view interactive map will take you to the spatial portal. So I'm going to click on that one there. All right, it's given us many views of the world, and we can just zoom. Oh my God. Okay, you'll notice that the toggle function in the spatial portal is a little bit more sensitive than on our species pages. So, zoom in. Cool. Great. So these are all of our taliquas that we have in the atlas, and they're all around Australia, which is really cool. But that doesn't necessarily tell us a lot of information. So I'll just point out a few things that you can get from here, and then we can always go back through them if there's stuff that you want more information on. But a really quick way to get a little bit more information is, just try to zoom. Sorry if this gets jumps around just a little bit. The map can be a bit difficult. So we've got this thing called facets. And if you click on that there, then you can sort of facet by lots of different things. So a really interesting thing to do when you have a genus is that you can facet by scientific name. And what that's going to do, oops, sorry, is just give it a second to load. It's quite a lot of data on this page. So when you facet by scientific name, it'll give you all of the different scientific names that are in this genus. And so some of them will be paired up and some of them will just be at the genus level. But you've got all these main ones up here. And as it loads, you'll see that there's quite a lot of information in here, and it's also quite visually appealing as well. So it can be just a really interesting thing and fun way to play around, which is great. Almost there on the loading. Perfect. So this is really cool. So now we've got in light green, let's see what that might be. Taliqua rugosa rugosa, and then dark green is sort of down right in Canberra over here, which is where we are today. And that's Taliqua nigrolata. And then we've also got lots of others. So that's really awesome. And if you were to click on one of them, it'll go back to the main color red, which is a little bit confusing, even though it says that it's blue here, but you've indicated that you just want this one. And so you can see that that's found all along the eastern coast and up north. And if you go with that next one and then untoggle that first one, you've got a different distribution. So that's really great, and that's always fun to play around with just if you want to explore some information. Other ways to get information is you can add to map, you can add a species this way. So you can add anything you might want. Let's go with the eucalypts there we go. So Eucalyptus pilearis, which is known as blackbutt. It's quite a common one. And you can scroll down and you can customize it and you can say, I just want Australia, and click next. And that'll import as a new layer. So let's change that color so everyone can see. Let's go with dark green. Actually, let's go with blue. You can also increase the size of these points and then also the opacity. So these two things aren't particularly related to each other, but they're just what popped into my head. So we've got a eucalypt that exists on the southeast coast, and then we've got this particular species of Taliqua. So if you go back to that one, then they overlap a little bit more. So these are just some of the things that you can play around with. There's also different sort of layers. That was a species, but you can add an area or a layer, and then you can also run some analysis. I'm not going to run through these right now because they can take a little bit of time because they're dealing with lots of information. But once you've got something and you have a question you want to ask or some information that you want, then you can go through and you can generate an area report, or you can calculate area of occupancy and extent of occurrence, which can be great. I'm just going to decline that right now. All right. Okay, just that last thing on our list is exploring your area. Now, this is a really common one. People really love to do this because you can get a lot of information about where you live. So I'm just going to go back to the home page here. And from here, I'll zoom in again so we can see. And we're going to go on search and analyze. So from search and analyze, we scroll down and we click on explore your area. Do that. That's not where we are. That's strange. There we go. All right, so we're in Acton in the ACT, in the office, the head office, and we've got all of the species that have been recorded in a 5-kilometer radius of us. So again, scroll around here. So there's a couple different things you can do to customize this. Obviously, put where you are, and then so 1/10. So we're going to go to 1 kilometer to make that just a little bit easier. And you'll scroll over here and you realize there's heaps of records that have been submitted around this area, which is really fun to see. So you've got lots of different sites. This is a very dense map of occurrence records. Scroll out a little bit and we'll go to the left. And now we have all this information. So in this 1 kilometer radius of sort of Acton ACT, we've got 3,543 species. And mostly a little more than half of them are going to be angiosperms, but also we have a lot of animals, and that's really awesome. So we can also filter by that. And if we go click on animals, you'll notice there's a lot of, yeah, so this is the amount of superb fairy wrens in the atlas, not in this specific area, but superb fairy wrens and different mammals. And you'll notice on the right here, I'll zoom back out so we can see everything, that map's going to change to reflect what we've clicked on. So just birds, just mammals, just insects. We don't have any bryophytes, couple algae records, some monocots. So from here as well, if you're interested, let's go to reptiles because we were looking at reptiles before. You can also then click on a record and then go back to that record. And now you're at a occurrence record page like we were of a record that was submitted.
Hi, Adam, it's Melanie from Beeline. I don't know.
Sorry, we'll just keep on with that. If you have any questions, just pop them in the chat and then they'll get passed to us as well. So now we're at a record from here in the ACT. This has a nice picture. We can see that as well. Got a nice lizard as well. So that's awesome. That's just an example of the way that you can sort of navigate information. You can find information about where you live and records that have been submitted. So that's the end of our tour of the atlas. And we've got some time for questions. So if you have specific questions, we'll go through the chat, and I might just stop sharing my screen and so that we can go through, and then I can reshare at specific points if we need.
It was great. Thank you, Margot. Awesome demonstration.
Right, hang on. I feel like I can't quite, let me just.
And we might also stop recording now, just so everyone can feel free to ask